I think you all grabbed some food, had a good breakfast, had a good evening yesterday. My name is Göran Hübner, once again from Bergen-Ingelheim, southern Germany. And it's a great pleasure for me to open up our third day, the last day of this year's Pharm conference, actually. But I promise you, we have a great program for you on our last day. So good morning, everyone. I can hardly see you because the lights are so bright, but I guess you're all there. Good morning. Oh, thanks for feedback. So it's a great honor and pleasure for me to introduce you to the next speaker, the speaker of our second keynote lecture, although I guess an introduction is probably hardly necessary. I first came across his name when I was doing a student internship in a mass spec laboratory. It must have been 2002, I guess. I have to apologize to this audience that it was not capillary electrophoresis at that time, but mass spectrometry. That was my first contact with bioanalytics. At that time, and also during my diploma thesis, I was to characterize like glycolipids, LPS, things like that. I was not into proteomics. But we had a high-res mass spectrometer, and it was due to a research proposal. After an upgrade, we even had electron capture dissociation, which brings me closer to Professor Kelleher. And we thought next idea was to use then capillary electrophoresis to separate our analytes, prior to mass spec analysis. If it took all this mixture and started searching for literature back then, you inevitably kept coming across his name. For example, I remember a science paper from '95 on atomole protein characterization by capillary electrophoresis mass spectrometry. That's something I still can't get managed in my lab today. And of course, also the paper by Roman Zubarev, Professor Kelleher, and Fred McLafferty in 1998 on electron capture dissociation on multiply charged protein cations. Well, I started working as one of the many small gear wheels in the machine of the pharmaceutical industry. Neil not only continued his already impressive academic career, but also spun out several successful companies. Nowadays, he is the Walter and Mary Glass Professor of Molecular Biosciences and Professor of Chemistry in the Weinberg College of Arts and Sciences. He's also the director of a 50-person proteomics center of excellence, the director of the Chemistry of Life Processes Institute, serve that, and a member of the Robert Lurie Comprehensive Cancer Center at the Northwestern University. And the morning I asked him how he's going to manage that, all of this. I can hardly run one lab with like 7 persons, but it's absolutely amazing. Up to now, he published more than 450 papers, including, for example, a 2011 Nature paper on mapping intact protein isoforms in discovery mode using top-down proteomics. He mentored over 50 PhD students, more than 200 postdoctoral scholars, and also more than 200 undergraduates. I hope these numbers are correct still. I mean, they keep growing. I could go on now for quite a while, listing his achievements, merits, and awards, but that would take up too much of his time to talk today. So I will stop now and ask you to give him a warm welcome. We look forward to learning more about Proteoform-Enabled workflows for robust characterization of biopharmaceuticals and endogenous antibodies. Professor Kelleher, thank you.
Well, thank you, Göran, and thank you for the invitation to come and the hospitality last night. As I got to my room, I had a little cheese plate that really was efficient, and as he just said, my life is requiring efficiency, and that includes this morning. So efficiency in conveying information to you. Kristen said we're going to melt the walls on this place this morning, and so to that end, I encourage questions throughout the talk. I think in a format like this, and in a community as tight-knit as this, we can break open the black boxes that we use. You'll see some new ones, and I think you'll see some excitement in the offing that should be translated into your labs or labs like yours. And we're going to also move what you classically would use for biopharmaceutical characterization, mainly MABs and AAVs, and I'll have some discussions about that using new technologies. But I also want to move into serology and endogenous antibodies, because the platforms we use are much the same. So good morning. Thanks for the visit to California, and I hope you find this interesting. So the proteoform promise is something I've been pitching for a lot of years. It is growing, that's good. And some of you who know me in an academic context and aligned with government resources, I'm pitching a human proteoform project, an arc and cadence of which would be like the Genome Project. So like we sequenced all the DNA in our bodies, can we domesticate, regularize, and sequence the proteome? I'm not going to talk about that today, but I welcome questions about it. And so I'm basically the pitch person for the value of assigning protein structure and composition with complete molecular specificity. So that's primary structure. And so, of course, for this crowd, we're doing antibodies. I'm going to tell you about some new tools, including that's used on endogenous antibodies. And I'll use the very same methods, and I'm sure some of you have heard about the charge detection technologies that have really grown up during the pandemic and in the days after. So I am a co-founder. Those are my disclosures. Many of the workflows we offer at Impro that you'll see today. Impro is a startup, it's a proteomics services company. And so you'll have an email later, but that is, these are available. And they actually link different things we've been working on, software and hardware called SampleStream, which I'll tell you about. So I'm going to give you a small dose about the promise of proteoforms and just the reality. This is really pretty simple, that the concept of a protein is not specific enough to capture our biology. It's not specific enough to capture the drugs, protein-based drugs that you're making and modifying with ADCs. Of course, you need to know the distribution of drugs and the DAR on those antibodies, right? It's the same concept, just brought to endogenous proteins that are created from the 20,300 human genes. Concept of a protein, it's not specific enough to capture our biology. So why should we think that drug development is going to be efficient? Why should we think that companion DX and biomarkers are going to be on point for proteins? We have to get molecular characterization with complete molecular specificity to make those be more efficient at enterprise scale. And that's it. It's the sum. It's not that complicated. And let me go back to like metabolomics as a field. Many of you are engaging with that field, right? You would never take a mixture of thousands of metabolites and start to disconnect the bonds between the atoms in those molecules as a mixture, and then get 20 or 30% of those atoms back and try to characterize the metabolites that were in that mix. That is describing bottom-up proteomics, right? So all we're saying here is like meet biology where it's created. And I'll have tons of examples of these and the value proposition that derives from doing that. And so in 2012, a group of about 50 academicians and some vendors, the picks and shovel companies, many of them are out here with you this morning. We went to Portugal and we were like, okay, so let's try and get serious about this, because the community of bottom-up proteomics was raging. The growth in the market was 30 to 40% a year. I mean, it was tremendous at this time. And so we said, okay, protein variants, charge variants, isoforms, PTMs, polymorphisms that are encoding regions or mutations, combinations of post-translational modifications, all the sources of variation in the primary sequence and structure of a protein. This is what the word proteoform was born to describe. It traces back to a single gene, and so there is no inference problem. There is no problem of making peptides fit into, it eradicates that, solves that, and so that's what it's meant to convey. And it's not just a word, it's a movement. And so it has a promise, and in biology, I'll just replace genes and traits, or disease traits and gene, there's this well-known genotype to phenotype gap. I'll just replace it here this morning with, protein mixtures and elevated traits that you're trying to correlate to, whether that's of efficacy or toxicity for your drug molecules. And so the assertion is if you can bridge those two worlds more efficiently in biopharma if you have complete molecular specificity. And this is the one area of biotechnology and life sciences research where I don't have to pitch that this hard. You all have been the market justifier for us in academics or in small startups. And it continues on that way. So I'll skip some of the biology. I already mentioned it briefly, and I'm gonna ski downhill as we go. But the other argument I'll make at the end of the talk is about drug target selection. That if you know the proteoforms of your target, oh, I don't know, let's pick one, tau in Alzheimer's, the protein tau, right? Apply what I'm saying now. Tau, that concept of a protein, it's not specific enough, right? Of course, you think about phospho-tau, okay? But I will assert, and now with some evidence that I won't show today, but there's over 800 proteoforms of tau. And they're quite different in Alzheimer's brain regions than control or healthy brain. And I'm asserting further that if you knew those proteoforms and how they changed over disease progression, that you would have new and smarter, better drug targets. If you knew there was a proteolytic activity that was creating fragmented tau and creating a hypothesized prion-like version of tau, you'd really want to know which protease that was. And you may even want to target it. These are the kinds of thoughts that I wanted to bring in general format today. And what I use in my academic life and in approaching the United States government, among other governments, to try and fund a large-scale project to make deep proteoform sequencing a reality. And so moving right along, all I'm saying is basic biochemistry, like in a signal transduction cascade that you target in oncology all the time, all these phosphorylations, right? You see them, but the proteoform knowledge is not there. I'll show you one example in the MAP kinase cascade that sits under, RAS and BRAF, the MAP kinase cascade. I'll show you one downstream kinase called MEK1, and rather than talk about it in general. But the idea is that if you can get that proteoform knowledge, it will correlate to function and phenotype very strongly, stronger than other types of molecular connectors that we have. That's my career, basically, in a soundbite. So here is, let me skip histone H4 being treated with a deacetylase inhibitor, right? So now you're going to get 42 Dalton, you're going to get hyperacetylation, which is what you see in these intact mass spectra. And then there's some methylation patterns underneath here as well. But let me just go right to MEK1. So, and I'll make a couple analytical points. So MEK1, it's underneath, it's in the MAP kinase cascade. That's its phosphorylation profile, and it's changing over time as you treat with MEK1 inhibitors or you get relapsed melanoma. And so many of you aren't necessarily geared up to interpret these spectra. These are large molecule mass spectra obtained by LC or CE, top-down MS. And so you can get the stoichiometry. If you just look at these profiles, in any spectrum, you can get the 1, 2, and 3, and there's a fourth. In relapse, you get a fourth phosphorylation proteoform there as well. 5 years ago, we did not have the tools to localize all those phosphorylations. Now we do. You get further in, and that's in this paper that just came out. And I encourage you to read about that, and I'll tell you about the underlying technology in just a bit. But the thing you get here is not just correlation to cellular or cancer phenotype in terms of aggression, doubling time, tumor size. You get stoichiometry. In a single spectrum, you get the relative amounts of 1, 2, 3, 4 phosphorylations. And these things that when you digest it, there's an English nursery rhyme. Humpty Dumpty sat on a wall and had a great fall, and all the king's horses couldn't put Humpty Dumpty back together again. That is bottom up. Even if you have 100% sequence coverage, you don't get to the same perspective as this gives you in a single spectrum. So this MEK1 story, there was 9 phosphorylations known in MEK1. There's also other MECs. There's MEC 1 through 6, right? So this is specifically this gene in the gene family, and you don't know which ones are active, which ones to target, which ones are present at what stoichiometry. It's very hard. You would have to create all those synthetic peptides and go through a whole lot of work, whereas you get that in a single spectrum. But right in that last point, I want to step back and say that belief that you can get orthogonal, and I'm arguing better information by top-down versus all the peptide madness that you would experience trying to localize those 9 phosphos, find out which ones are the functional ones, which ones are the high stoichiometry ones. This is the balance, but people think, oh, top-down's too hard. It's, the tools aren't there, right? And that, 10 years ago was true. Every year that goes by, that's getting less true. So it may start to tip your balance in your own organizations about who to hire, what platforms to invest in. And so here's now, I slow down the video. Here's MEC 1 in a default cell line. So this is just a cell line model, super simple. Doubling time is there. You treat with a MEC 1, a BRAF inhibitor, vemurafenib. Probably many of you know it. Look at that. There's the 1, 2, and 3 phosphos. Look at the 1 phospho now. Used to be the most abundant. Now it's second to least abundant. So these are what I'm talking about, correlations of these profiles to cellular phenotype. And then here's a relapse model. Look at the 4. This is what I call an angry proteoform profile. I do not want this in my body as a melanoma. That is a hyperphosphorylated state, and usually people are gone within 3 to 6 months if they get these kinds of relapse mutations. Okay, so to CE and to the topic at hand, these are all ways in which this community knows why this technology is valuable. And indeed, in the Genome Project, it was capillary gel electrophoresis with 96 capillaries, right? That's how we mapped and sequenced the genome. And so I have this vague hope that we can get the band back together and do that same thing. I just saw 8 capillaries out at the SciEx booth. And so, these things, you can start to see how to scale this kind of approach where it's tiny sample consumption. And now we've got both in denatured and native mode CZE, and you'll see examples of both. So I won't belabor this. About 8 years ago, we got 2 of these instruments from SciEx. Back then, they were called the CESI 8000s. Now they're the PA 800s, I gather. And so we interface them to the Thermo world. So we do denatured on the tribrid platforms, and we do native on the UHMR, and we had a hand in co-creating that instrument and launching it in 2018. And then all of our software, a lot of it's commercialized either through Thermo or through Proteoform-Enabled, a startup company. And that's how we get our proteoforms out, is that software is a key element. But it's not academic hackware. It has been out in the commercial private sector for 15 years or so in the form of ProCyte. Okay, so let's just set the table, starting simply. Here's a postdoc, Kevin Joos, who's now got his own independent lab in the Netherlands. He was doing some basic optimizations of the BGE, shrinking the amount of acid, getting tighter peak widths. These are probably things you've known or even seen this week, but some charge variants here are now detectable, and they can be mapped. So these are intact, just on NIST standard, just beautiful peaks. Peak width is tight. This is a Q exactive, and you get the glycoproteoform landscape there, and you do the same for the charge variants in this case with 1 extra lysine. These are all standard things. Most of what I'm going to say for the endogenous antibodies utilizes all what is known to you, IDES digestion or just reduction. But with IDES, of course, you get the 3 pieces that are nice in this middle down size range, et cetera. And I'm not going to belabor this because you all know it. So in a CZE top down, middle down experiment. Let me show you some fragmentation data. I think intacts are very well known to the community. Doing actual fragmentation and getting to the fragment ions, that's the step that I think still there's trepidation about. And, but the tools are getting there. So here's if you did that on the FD or the FC and the light chain from an IDES digestion, you know, there's the intact, and this is on the fly using a tribrid mass spectrometer. In this case, it was the Eclipse. There's the fragmentation patterns, and so what do you do with that, right? Those are fragment ions that go up to 20 kilodalton fragment ions. And so here's the interpretation of it, and this is routine, robust stuff. And now, you know, this is not optimized, just rip it through. Here's the amount of evidence that you can expect, I think actually expect, in this maturing method to localize, for example, the N-glycosylation site, or other sources of variance. And you look at this, and I had a lot of feedback 10, 15 years ago. It's like, oh, well, you're not getting this, and you're not getting that. You're not getting localization, right? You want single amino acid localization power if you have a modification or you have drugs bound to an antibody. And that's true, but now on the tribrids, you have 4 different fragmentation methods to choose from. And if you unleash them on this, you now can get 60, 70, 80% coverage. And there's academic labs like Luca Fornelli at Oklahoma who's showing 90% sequence coverage. And by that, it's meaning to say that these blue flags, that's the amount, that's where you get a fragment ion that mapped to the cleavage of the proteoform at that spot. So now you can imagine getting 80, 70, 80, 90% coverage at this level, and then you've solved that problem. That's one way in which this field is maturing pretty rapidly, and that's at 25 kilodalton, right in the size range of all of these pieces.
Neil, may I ask a question?
Oh, please.
Because you asked us to ask.
Chair's prerogative, yeah.
What's the peak width required? You know, I guess usually, you know, those colleagues are using ALC chromatography before with rather broad peaks. Because, you know, coupling or combining these different fragmentation techniques and getting to like 90% sequence coverage will take some time, I assume. What's like the peak width that you think is feasible getting such good sequence coverage? Because, you know, with coupling also CE-based techniques to mass spec, maybe it's interesting for us nowadays to do ICI-FMS, and the peaks are not like minutes wide, not seconds wide, but something in between. Do you think that this would also apply, or that it would be possible to get in these ranges with a CE-based assay?
Yeah, so this is CE. I didn't, yep, this is capillary electrophoresis, so you might not be impressed with, yeah, 30 seconds is, you know, okay, I'm at the...
I mean, it's good for mass spec analysis.
Don't storm the stage. You know, I'm sure Kevin could have done better, but we'll blame Kevin. I just threw Kevin under the bus, yeah. But I think he actually detuned it for exactly your reason. He was trying to get more time, to get more fragment ions, and that's, you know, because we get this criticism a lot. This is also 2020. You know, Kevin was in the lab 2019 through 2022, and yeah, so I think it's tunable, and that in current speed, you know, now there's the Ascend. You know, they're always trying to make these things faster, but that development, at least at Thermo Fisher Scientific, it's really geared around bottom up, and you know the whole Timms-Toff, or you know, the speed race now at whatever 150 hertz. But that speed has assisted this. And that's part of what I was saying, that you can get 70, 80 on an LC combined with CZE, you know, detuned CZE. You could get that sequence coverage today with commercial tools. That is not an overstatement. And even with these sort of peak widths. So it is true, though, in all the different modes of capillary-based electrophoretic separations, you can get incredibly tight peak widths. So, and that can be too fast still. So yeah, keep the questions coming, because understanding is the main thing I want you to take away from today. So Let's just reduce an antibody. There we go. We got light and heavy. So now I'm going to really change something. You may not have heard about this before because a tree fell in a pandemic and nobody heard it. Tough crowd here this morning. Yeah, that was so those who do intact or middle down, and there's your light and heavy chain. Very honest spectrum. Any old day, there's some adducts on this. There's some guanine adducts and things, and just I'm not overselling this. So that's tough to deal with, right? And sensitivity, sequence, coverage, etc. So what I want to say now is that now is turned to this, which it's the same sample, same instrument, it's just shifted modes. And now look at I've changed the scale, mass to charge to mass. This is now called what Lithermo calls direct mass technology. But when we invented it, we called it I2MS, or individual ion mass spectrometry. It's a fundamental shift in the readout of protein mass. Okay, and that's part of this shift into the new tools that I'm going to talk about that now have been commercialized. And I'm going to focus a lot of it on endogenous antibodies. And that brings us into the world of serology, something we just lived together. So there's your dreaded control and your dreaded true. You now have COVID. Great. So that's from an IgG titer, right? That's from IgGs. This is a perfect audience to talk about this. What is the molecular pattern if you could have single proteoform resolution? What would it look like under that? All these different IgGs. IgGs, so many versions of them, right? Well, what had happened here? You put an antigen on the LFA, the lateral flow assay. You put an antigen, probably the spike protein of the virus, to attract the IgGs. So that's this experiment. So now I want you to think about what the IgG, could you resolve single clones? Single B-cell clones from a sample like that, treated like that, where you do an affinity capture, just like this, and then read it out if you could have single ion mass spectrometry. And it looks like that. There's your kappa and lambda. This is just the light chain. This is after reduction. So this is not the intact IgGs. This is just the light chain after reduction. And that's an internal standard, so it's a clinical grade assay. It's a known sequence at 50 nanograms spiked in. And so there's your pattern. And you can resolve single clones and assess different people. This is person 1, person 2. So you can get not just a titer, but how many different clones somebody has generated to fight off an antigen. And I'm going to slow this down, tell you how I got the data, and then show you some more patient data as we go. So what we did over the pandemic was to integrate a fully automated system to do this for a clinical grade assay. And so we had to stitch together many components. We had to use a Kingfisher for automated immunoprecipitation. This device I'll tell you all about because it's a startup company of mine, Sample Stream. That is the concentration and buffer exchange device that sits in front of this technology I2MS that I'll tell you about to then generate the data. And this is a really severe COVID patient where the spike in at 50 nanograms, you can see they have very high titers, lots of different clones. And so we had to create some new metrics to track that. Right, okay, and that's been published in 3 different papers so far. Okay, so let's break it down. The first part is, as you might expect, take someone's serum or plasma, put it on a solid support, take 100 microliters of plasma. We're down to 10 to 20 microliters of plasma or serum. And I think we can get to dried blood spots, but we're not there yet. And in that, the IgGs will bind to the spike protein, the same construct, 30 kilodalton construct that everybody uses for affinity capture of the IgGs. Elute, create heavy and light, and then use this new readout. And here's the fully integrated system that we built during the pandemic and controlled airflow in there because blood is what's going in here. Excuse me, serum or plasma is what's going in there. Okay, and so there's an example. So I'm just showing you that light chain from a really sick person there, and we'll break it down as we go. Here's example. Well, I'll come back to it. Let me first show you how the data are obtained. But you can see clear differences between severe COVID on your left, 2 people with severe COVID, and 2 people with very light COVID. Okay, so the first step about once it comes off the Kingfisher after automated immunoprecipitation, it goes into this device, which is really the brainchild of Phil Compton, who got his PhD from Don Hunt in 3 years. I couldn't believe that when I said I had to have this person as a postdoc. 3 years after that, he co-founded this company, Integrated Protein Technologies, to do this idea, which is to create a robust, integrated system. It's a PAL3 robot from CTC, and there's the sample stream module, and in there is a 10 microliter flow channel that does this. It's a form of chromatofocusing that uses 2 opposing liquid flows at 50, 100 microliters or a minute, and it creates this concentration effect where above a molecular weight cutoff membrane here, you get there's downward flow, but the push opposing forces of the flow create a very focused analyte band and allow you to buffer exchange as well. So it's a concentration and buffer exchange device. It is meant to compete with things like rapid fire, or in some cases, flow injection analysis, or LC. And so after you do this focusing step, which can take seconds or a minute, and you can inject arbitrary large volumes of dilute sample into it, then you block one valve and you mobilize back through the membrane and back to the mass spec or to a fraction collection tray, like right here on the CTC. So that's sample stream, and I just went through this, so I don't need to tell you, but at least I can show you. If your eyeball is coming in this way, that's what you see. Buffer going through, there's your molecular weight cutoff. It's basically like those spin filters, those little spin filters everybody uses and loses sample on? Yeah, it's a industrialized version of that. Very simple. Okay, moving on. And out comes a chromatographic or electrophoretic-like looking peak. And it's a laminar flow on the way to the mass spec, so you get what you might imagine. And there's all sorts of other things. It's all on the Integrated Protein Technologies website. That's all what I just said. Phil's been doing this for 8 years. This started in 2012, so this thing's come a long way since its beginning. So once this goes to the mass spectrometer, you got a peak there, right? The change I'm going to do on you is this new type of technology, the I2MS. And I gave you a hint about it earlier, which is it's no longer a mass spectrometer. A mass spectrometer is actually a Thomsonometer. J.J. Thomson, 1901, that's bad. That's a really bad, geeky joke. But it's meant to say that it's moving from a Thomsonometer, it's not a mass spectrometer, to an actual mass spectrometer, something that spits out mass. That's what it's supposed to be, right? So here's now a 4-protein mixture, all the usual suspects, what you might expect. There it is as a mixture, right? And this is what, if you get a really complex mixture for electrospray, denaturing electrospray, this starts to turn into a hump, and you can sort of see it becoming that way, which is why electrophoresis with separation of things was so critical for a long period of time, and why we got into it for top-down. But now I want to take the same mixture, and there it is. It's in the mass domain. You now have a much flatter baseline, and let me tell you how this thing works. But it directly spits out mass above 6+, so it has a floor about 5 kilodaltons, and that's because it uses charge detection. And this is something that AAV people know a lot about. Maybe you've even used some of the services at Megadalton. What it uses is an Orbitrap-based, multiplexed charge detection. That's what it is. And unlike Evan Williams or Martin Gerald, they've been working on these ion traps, which is true single ion in the trap, right? Moving around, it's got frequency and precision, etc. We did this on an Orbitrap where it was known in 2009 that you could detect a single ion. What you couldn't do is assign the charge to that single ion, and that's what we fixed. And here it is. in use of native, so this is native mass spectrometry, native electrospray. A tetramer is shown here, standard charge state distribution, with an indication of how signal is generated in Fourier transform mass spectrometry. So the basis of signal is constructive interference of the ions in the trap. So you load up the trap, maybe you have 10,000 ions. The frequencies are all over the place, but when they constructively interfere, you get peaks. And that's your genesis of signal. No longer. Now, if we dilute the sample by 500 to 1,000-fold, that's the first thing we had to do, is take the sample and go from 5 micromolar to 5 nanomolar, because we were after just single ions. And let me take these off so you can see. This is 50 ions, okay, from that same exact mixture. And you can see the high abundance charge states, you get multiple isotope peaks, and the low abundance charge state, like the 46, you get a single peak, right? So probabilistic things are in effect here as you go down to the single ion limit. But now I have to take a single ion, which I know the frequency incredibly well in FTMS, in an Orbitrap. I know that frequency like 1 part in 10 to the 8 at least. It's just locked right on the frequency domain. And so I can follow it, and I can integrate the signal of that single ion over a 0.5 to 2 second detection period. So that's what these detection times are. And that slope turns out to be proportional to the charge. Okay, so if I have a single ion in the Orbitrap, which I have 50, right? But if I just track 1, that slope is the charge state of that ion. We just had to calibrate it. So if it's a 49 plus, like the one up here, that one's going to have a slightly higher slope. If it's 47 plus, 46 plus, it's going to have a slightly lower slope, like this. So this is now, this is all published now. We broke this open in about 2018, and a tree fell in the pandemic, and maybe a few people heard it, but it really changed the pace at what we can do with our science because of these advantages. Not only do you get the 500 to 1,000-fold greater concentration sensitivity, you deal with it. You can handle complexity, and you'll see examples of that, because the basis of signal is no longer the constructive interference. It's every ion is the basis of its signal. So it doesn't matter how complex the mixture is. And when you're dealing with a single ion in its position, you actually get a 10 to 20-fold boost in the resolving power that you achieve, because you know it's a single ion. You know its position. So you get a resolving power boost. And here's the 40-second video showing 4 ions in an Orbitrap doing this. There's the current, the story plots, we call them. There's the frequency domain. And you just take the frequency, which is mass to charge, times the charge, giving you a mass spectrum. And we can do up to 20,000 ions at a time, but typically it's like 2,000 to 3,000 ions at a time. And that'll become important when we get to AAVs. Okay, so we're going to go back to this serology. Now you might be able to piece together how we're taking 20 microliters of serum, doing an immunoenrichment, finding the IgGs that bind, release, digest, we're doing this, so we're just reducing in denaturing, and you folks know all about that. And now this is the light chain region with all those advantages that I described. That's how I'm getting this data, which really, this mixture, and these are single clones. These are reflective of single B cell clones in the light chain region. And we also have data where people were actually on monoclonal therapeutics, and there'd be like, what's that thing? Oh, it's a monoclonal variant, and that has many implications, but not just in COVID, but across oncology, as you might imagine. So we had to create metrics for like the degree of clonality, like how many clones. And when you see much older people, they get less clones, right? They have less clonality, different B cell clones. We see anecdotal trends of that. We haven't done a detailed study design, but there's all sorts of potential uses for this method. And yeah. Yeah, and that should be here. So do I have non-COVID? Well, non-COVID basically looks like this very light COVID patient. It was an outpatient. Yeah.
I meant long COVID.
Oh, long COVID. Ah.
Because I think that's probably even a more severe disease these days.
Yeah. We stopped. We didn't get that grant, and I don't have data. There were a lot of grant money flying around, as you may remember. Yeah, I'm not asking you for money, although, are you? Let's talk afterwards. Right, long COVID. That would no is the simple answer, but and this thing keeps auto advancing. But right, what I can show you is the deep COVID. So this is like someone with 3 weeks, in the hospital, with obviously more clones and more titer, high titer, which is known, right? High titer for if you have severe COVID and your immune system's battling that off. Whereas people that were, these were between 1 and 2 weeks, and these were more 3 to 4 weeks. So it's not apples to apples. Right, it would be very interesting to look at long COVID. So that's about where the lab stopped and pivoted and started to, we are interested in sequencing those clones, absolutely, right? We're technology developers. Yeah. Oh.
Can you do peptide mapping after this?
So if you took a mixture here, you have the mixture of light chain and heavy chain. And I'm not showing you heavy chain for time constraints, but if you were to digest this, you have now the inference problem. So you see, if you were to you lose the connectivity of what all those light chain clones are. And then you have this inference problem of trying to reassemble the peptides, CDR or conserved region peptides, into, even if you had this information, you would have a giant informatics challenge on your hands. So I would say no, if your goal was to try to explain the sequence of these different clones. You have to do it by tandem top-down MS at this level. And that's what we're working on. But I'd love to, if you disagree with that, or, let's, because that is exactly the point of where people all the time are just going to digest, right? You've got these labs, and they're just seething, bottom-up, incredible organizations that you guys have, and you just use it, right? Put it through the sawmill. Let's chop it up. I'm like, when you do that, you have artifacts that are introduced, you have all sorts. Yeah, okay, I'll stop. But Tim's going like, okay, you okay? All right. Yes, with some of these services, because they are hard to set up in your own organizations, right? If you were to pivot and say, oh yeah, let me hire 2 former people from top-down labs, and then get new hardware, and you go to your management and say, yeah, let's do this, like, that's $1 million. And that's maybe not where you want to spend $1 million. So if you're in that position, Jack McGee was with me for several years and now spun out this company. It's a services company. It's meant for exactly that moment. So if you want to dabble toes or use some of these assays that use this daisy chain of sample stream with I2MS, direct mass technology, and then the readouts in a standardized report, Impro is the place to go. Yeah, so that's exactly what Jack sent me here to say, and so I'm here to say it. And yeah, I think it could be a nice market creation, market development engine, as well as providing initial value points and value assessments out to the industry at large, i.e. you all. Okay, so let's use this same integrated platform for AAVs and see how it comes out. So now we're going to go to much higher mass. We're going to go to the native, and this is now a system that uses the UHMR type Orbitrap. And what we're trying to do is get around this, all manual data acquisition, where you've got your staff member with these little needles doing native electrospray at low flow rate manually to get out and use, for example, single ion mass spectrometry in the ion trap or Orbitrap, right? That's what that looks like now. And it's not widely accessible or widely known. And so what we did is automate. We used the same daisy chain to connect them together and get this all automated in less than 20-minute run times, and that's what Sam has done in the lab. So I won't take you all through, but basically you get empty versus loaded, or empty versus full capsids, et cetera, and it uses the single ion MS. And we did it with just standard electrospray source, a Hezi source at a few microliters a minute. It can improve by using lower flow, but that gets harder to automate, right? So we have got an automated low flow system, and we're still working out the kinks. But this is the source, and here's the 10 samples all in a row, and I'll even show you some from media as well that can do this kind of high, reasonable throughput for this AAV analysis. That's time in hours here. So here's some of the data we're seeing, empty versus full, all sorts of ratios, the actual mass of empty versus filled. Here they are, and the theoretical is about 0.8 megadaltons or 800 kilodaltons for the full. We're seeing slightly higher than that, slightly higher than 800 kilodaltons, depending on which AAV is used. But I'm moving pretty quickly through this, so please ask a question if you like. Here's when we have empty versus full, the reproducibility of it is quite good, and we contrive these mixtures. We create different ratios, 50-50, 70-30, 30-70, et cetera. And I think that's shown on the next slide. Oops, no, I took it out. Okay.
Are you able to see any partial fill-in?
Yeah, so, and double filled. So I think if we go back, you see that's double. And the partials, in this higher flow rate regime, we kind of obliterate that partial. I think that's due to partial filling as well as lack of desolvation or other things I have to wave hands about. And we're automating this report, so we don't, and the whole system was, automation robustness was the top goal, and so we didn't stop to optimize that. But we have seen it, if you go really low flow rate, you can start to see those depending on which AAV. But we do see robustly the double filled. Yeah, sorry, I had okay, that's right.
Hey, Neil,
Yeah.
how easy
Yeah.
can you do this? Like the automated method,
Yep.
can a John Q. Public do it, or do you need to be able to run a nano-L? I mean, yes, it's not nano-LC, but I've been owned by a nano-LC thinking that I can do mass spec. So how?
Wait, don't you own the Nano LC? It doesn't own you?
Just thinking, because we're on the cusp to potentially have, MAM is a thing now in biopharma, or at least trying to be more of a thing for our friends at the regulatory agencies. And I know empty to full is a tricky slope to do. So I'm just wondering, because it's all well and good when you're academic, you have grad students, this is their life, they can
Yep.
figure it out. But then when
Yep.
you try to roll it out to, let's say, a dev lab that has a specialist, could it then go to a GMP lab that maybe the specialty isn't there, it's someone else, but then QC, could it potentially go there?
It was designed for that. This was a joint collaboration with Thermo, launched a year ago, and that's why we did everything we did. And so there's an academic paper coming out on it. But really, I mean, it was designed to go, here, right? I mean, that's translation in academics, and so that's the spirit of my answer, is to not oversell and say that is what it was designed to do. The components of it are industrial grade, so the UHMR that this thing's attached to, that's all you need. And the methods, each of this is has LC-based methods in the sample stream unit that are robust, that are, so that was what is exactly, for that question, I did not seed her with that question. But that is a great question, because you've seen academics for years roll through here and like, yeah, we did it once, and the postdoc got a job, and now they work for you. So if you send a note to Jack at Impro and say, look, we have 3 or 4 customers, and let's do it. Let's test this. And the number, and then the question is translation to your own labs. Do you want to do it? Was really what you were asking. So yeah, UHMR sample stream. The methods might take a month or so to get this kind of consistency, where that's a different sample. And he's done 100 of these in a row, and that'll be in the paper. Okay, great. I love the questions throughout. I think that's critical for a morning as I go through a lot of material. And yeah, so let me switch back to CZE and show you an application that you may not have seen coming. It's not antibodies. It's nucleosomes, endogenous nucleosomes, and how CZE was able to explain what is actually in the mixture. From what you may know and from your chromatin epigenetics and cancer epigenetic programs, which I'm sure many of your drug companies have, are targeting chromatin remodeling enzymes. You know about chromatin biology, where you have 4 histones, 2 copies each, in this nucleosome structure. And this will be important for what CZE resolved, is these ragged ends. This should be 147 base pairs wrapped around 8 proteins. But actually, it could be 148, it could be 149, it could be 150. The enzyme that cleaves here can leave these ragged ends. And so that's the punchline. So this was Kevin, who I threw under the bus earlier, but he's strong enough to take it. And this was with, again, the CESI 8000, and EpiCypher is a reagents company out of North Carolina in the epigenetic space. So first, what Kevin did in the lab was take that CESI 8000 and develop a standard method for native top-down analysis. And so rather than take you all through it, you can imagine what he did and what all the conditions are. This was published some years ago. Let me just show it in action. On native carbonic, it's keeping the zinc because we're in native mode. Even for a pyruvic kinase, 280 kilodalton tetramer, you get nice separations and even some minor variants of the tetramer, where one subunit is cleaved slightly, changing its mass once and twice. There's two of the 4 subunits are so truncated. This is all published, so I'm not belaboring it on a Wednesday morning. But Kevin did a great job on this, and so it's really detailed method paper. So now let's use it for nucleosomes. And what we're going to do is what we call nuke-MS. That is the process of taking an intact nucleosome, 200 kilodaltons, and doing controlled disassembly, doing top-down of it, to eject out the subunits in an MS2 event, and all 4 subunits will come popping out of this complex. And we've shown this on 200 protein complexes from cells that we were natively separating as complexes. And that was in a 2018 paper. But a couple of years later, I had a student show that this was possible, to take the complex of a nucleosome, eject out the 4 histones, and then fragment the ejected histone proteoforms and get site-specific PTM information in a pseudo MS3 event. And so this is the paper that came out of this, 2021. There's Kevin again. And this really blew my mind. So EpiCypher creates synthetic nucleosomes, right? And they have them in a really nasty matrix. So here's the storage buffer. Kevin says, I'm going to eject that directly. And this is the electropherogram he got out. And I was like, wow. Anionic separation, there's the salt that comes out, and they're beautifully clean, are the nucleosomes. separate it out from all those small molecule charge contaminants. Of course, it's electrophoresis, right? And then the sensitivity of the Cesi 8000, if you probably know, the flow rate is exquisitely low out of this thing. And it elevates the sensitivity for intact analysis. And the desolvation's beautiful. I mean, yeah, this was, I was very impressed by this. And then ejection out of, let's do some standards. So EpiCypher can create synthetic standards where they control the PTM placements on the histones. So here's a mixture. So we're showing off electrophoresis resolution here. Here's a monoubiquitinated, so it's a whole nucleosome, 200 kilodalton with one extra ubiquitin on it, versus one that has triacetylation on H3, histone H3, lysine 4, 9, and tetraacetylation. And he can barely separate those two, but then he changes conditions and he actually can get almost baseline separation and just beautiful spectra to come out on the UHMR. So now we're going to do the full experiment, and now we're taking a whole cell line. We're taking 10 to the 8 cells. We're isolating the nuclei. We're isolating the nucleosomes, mononucleosomes, because we're treating with an enzyme called MNase, and that's clipping all of the nucleosomes, which are strung together normally. We're clipping the DNA, and we're creating mononucleosomes from cells. We don't even know how many proteoforms of histones there are, much less how many nucleoforms there are. I just used a new word on you, nucleoforms. Yeah, it's got to be over 10,000 different forms. That each cell has about 100 million copies of a nucleosome in it, and so there's plenty to go around. But I'm just going to show you spectra from zone 1, 2, and 3. And so there's 1, 2, and 3. And what Kevin found is that that they separate by a base pair. And because this is duplex DNA wrapped around this, right? So if I have an extra base, this ragged end I told you about, there's actually going to be a base pair as a ragged end here. And so that's what is creating all this heterogeneity, is the DNA, which is not necessarily what we wanted out of the experiment. We wanted to get after the histone modifications. And so for that, we eject out, we do nuke MS, that is to say, we eject out of this complex, and we can quantify. So here he's showing 2 different cell lines and quantitating what is known biology, that in this TKO cell line, they have less methylation equivalent on histone H3. So histone H3 is the most modified core histone, and it's got this whole methylation pattern. Each of these is 14 Daltons different due to a combination of methylation and acetylation. But anyway, long story short, and please ask questions because I literally flew through that story really quickly. But it's known that TKO has less methylation than NTKO here in blue. So the blue is shifted a little bit higher in methylation equivalence because it has an upregulated methyltransferase for histone H3, lysine 36. So it's getting way more dimethylation on H3 from this cell line. It's a well-known model of multiple myeloma. That was really fast, so if you want to back up. But it just really shows the power of the separation power. I mean, sending it a mixture like this, it was a little bit of a crazy experiment, but Kevin got it to work. And, it didn't work out the way we wanted, but we were able to get out histone proteoforms and even fragment some of these proteoforms in the MS3 event. Okay, so how are we doing there, Goran?
50 minutes left.
15?
15.
Good, 50 minutes left. Excellent.
I'll have questions in between. Don't be shy.
Okay. Yeah. So I hope what you saw in there. I hope it was worth getting up this morning and seeing some of the new, what a crazy academician goes off and does with your great tax dollars, spending on research, trying to build better tools. I said I'd come back to some of this, and at the end, the promise of the proteoform, I hope it lives up to this. The thing I said I'd come back to is this, and I think this may start to speak to our overlords. Everybody has a boss, right? And in drug development and discovery, let's go back to the source. So why do I think proteoform analysis is worth a $3 to $4 billion project? Speaking of your tax dollars, that would come from the US government or a coalition of governments. Like in the Genome Project, what could justify that? Why would we want to create the abilities to do deep proteoform sequencing and discovery and resequencing, just like we did when we mapped the human genome? We got the reference human genome. Now, 8 years later, here comes NGS, right? Next-gen sequencing. They couldn't have done that. They couldn't have done the 1,000 cancer genomes and modern genomics without doing the tech right first, and that was the Human Genome Project. So I'm arguing for the same thing, but to domesticate protein-level biology for the benefit of all, and let's just focus on one element of that, and the selection of drug targets. So, kinases have been targeted, the kinome, of course, that's been targeted, but we don't know the effect at the proteoform level. So as a consequence, what I'm arguing is that using a combination of bottom-up proteomics and antibodies, phosphoproteomics, that we really don't have a clear, efficient way to sort 50 drug candidates for a target. Which one's operating better? Which one's informing the drug discovery industry? That's one type of argument where you've already chosen the target. But maybe if you could use a variety of interrupters, RNAi or CRISPR, and you knew what I showed you on MEC1 before. If you knew the effect at the proteoform level on the cascade that you're targeting, question, would you actually select a different target? Would proteoform knowledge inform drug target selection? I also gave you a tau scenario at the very outset of the talk where proteolytic action on a protein like tau, which is absolutely active in the Alzheimer's brain, creating different isoforms of tau, even beyond the ones you've heard of. You're getting a really complex proteoform landscape. You're literally falling apart if you're tau through the course of disease. And I don't care what part of the region of the brain, even the frontal cortex, we see that. And so what are all those different fragments? They are reasonably reproducible. So what are they? We're still in the process of mapping them, but maybe the knowledge of them through disease progression and in the order of events, it would lead you to a different choice of drug target in an industry that's, let's face it, in need of prioritization of good drug targets. So this is the sort of thing that, I'm an academician. I'm not in the drug discovery industry, but there's a lot of logic here, and it's a daisy chain of logic that needs buy-in, needs community. I'm very interested in your thoughts about that. If we are going to get community buy-in, we have to discuss it, because then you could get the technology that, yes, it's hard to get proteoforms. Yes, you have to have a lab dedicated to it to really be able to clear the fog and then get deep proteoform knowledge, like what we're getting on tau. And so this is why it's a foggy, difficult conversation. But the technology, I would assert to you today that we are closer to the Human Proteoform Project, the distance in technology between now and the 10 years from now, if we started it today, that would close out the project, than genomics was in 1989, when those courageous few stood up and said, we need to sequence the entire genome. That was a big leap in technology. And some of you saw today why the basis for that statement. So I know that's provocative, not always the best way to land, but I did it. So I wanted to thank Jack. He's the COO of Impro now. Kevin did a great job. I'll tell him I threw him under the bus, and he'll laugh. And Sam, he did that AAV. And I will say to your question earlier that he did not know mass spectrometry. He was a first-year postdoc who did bioinorganic chemistry. That's Sam, and he did it in 1 year, what you saw today. So with that, I want to thank collaborators and EpiCypher as well, who's not on that slide, but most of all you, thank you for the invitation. I hope you walk away with some new knowledge and maybe some challenged perceptions. Thank you.
Thank you so much for a really inspiring talk, and maybe more questions coming up now. We're more used to discussing in the end.
Yep.
But I was glad that we had a few questions already in between. So we have like 10 to 15 minutes left for questions. So, please.
Come up to the stand if you like.
You want to take a seat or stand?
Right. Thanks for the very, very exciting topic on proteoform. I'm Ryan Nye from CSL. I was wondering for the key challenge, one of your number three, the third topic that you talk about. I was wondering if any mRNA vaccine companies actually approach your lab to try to understand, because they deliver a single gene, but as you alluded that there are different proteoforms, which would, I would say, it will give rise to different kind of antigenicity due to those modification as it expressed in human cells, right? Because in pharma, we actually able to control those monoclonal and then study those monoclonal antibodies and also even vaccines. But I anticipate that mRNA would have the challenge that I think your technology would be able to address or help them to elucidate the different kind of antigenesis response. What's your thoughts on that in terms of how your work and link to mRNA vaccines?
Hey, 3 thoughts come to mind. Number 1, there's a bunch of RNA, a direct analysis of RNA, and big ones, the stuff that goes in to the vaccine formulation. Like there's a lot of, like Moderna's going pretty crazy on that, and like 1.8 kb worth of RNA and flying it by intact mass specs. That was one thought. Second thought is the direct answer about what proteoforms, like sometimes there's glycosylation on some of these epitopes encoded, and like, so what's the glycoproteoform distribution that's sitting in, okay, I just got a shot, and you know, is the technology up for it? I believe it is, but you would have to have an immunoenrichment of the encoded protein, so you need a really good antibody or other affinity reagent to pull out. And now with this trace level, I mean, we can handle sub-nanogram amounts of material, and so I believe it to be possible. And what was the third angle? I forget exactly. Well, it may come to me, but I think I don't know that that's been done yet, Ryan. But I think it's possible to do, which would be really interesting to look at efficacy. And oh, that was my third thought. Here's how to look at efficacy of the vaccine. If you had a vaccine, I would kind of want it to look like the left side if it's efficacious, so the time scales for vaccine development could shrink. Because I'm saying, like aligned with my central message today, that these patterns reflect an overall phenotype. And so if a vaccine's really eliciting, and we actually published this, we published that that's a severe COVID person, but the vaccine second dose looks a lot like the red on the left. That's an effective vaccine. So yeah, those are my 3 thoughts.
A very nice talk, thank you. You mentioned, you showed some results when you did the MEC1 test with a cancer cell line. I was wondering if you have done the same test with real cancer tissue?
Yeah, that's a great question. And no, so those were cell lines, and I'd love to get melanoma tissue do the same. It's the same platform here, by the way, that the immunoenrichment of MEK1, we would need about 50 milligrams of melanoma resected tissue, and I'd love for that to be someone on the drug vemurafenib or everafenib or um, any of those other IBs. I can't find that MEK1, but yeah, oh, there it is.
Yeah, this is.
Yeah, it's cell lines. You got me. Yeah, I mean, cancer docs. The tissue is the issue, right? Yeah, Jenny Van Eyck, the tissue is the issue. I don't know if you have a study design with 10 patients in different categories, but if you do, let's talk.
Thank you.
She wants to test how much these patterns link to phenotype, right? Is the patient responsive to the drug? Is the patient, so it would look like that middle spectrum, or they're in relapse and the drug's like, you're not looking good. That's what I really want to test, right, yes, great question.
Hi. As a separation scientist, it hurts to make a separation worse for the mass spec. We've seen, particularly with top-down, we've seen the instrumentation progress a lot, but it is still quite slow. Do you feel like we're close to getting to a point where we can do our very efficient, very fast CE separations into a mass spectrometer and take advantage of all those top-down
Yes, so
fragmentation techniques?
that's a great question. Make it faster, and it's not. So, where's that peak there going? There it is. Yeah, detune that, right? Yeah, great. If you have a 5-second wide peak, that's going to be tough because the unit of currency in middle down, top down is the number of fragment ions that you can observe per unit time, right? And the instruments have been optimized for bottom up. The methods have been optimized for bottom up. We as a community, especially when you're in grad school, or the training that most of the HR pipeline get is speed for peptides. So that has an indirect effect towards top down. They have become faster. And I use thermo language because that's what I'm most familiar with. But as you go from the Lumos to the Eclipse to the Ascend, it is getting faster. There's something called top speed, and you can do a top 10 or top 12 type of data dependent experiment. But each scan is not going to be 50 or 10 milliseconds. You just need more ion current. And so it's more like 100 milliseconds. And so until we solve that, but if I'm a vendor, I'm creating instruments around the marketplace. The biopharma speaks with the largest voice for top down, middle down that there is. And so, but they also try to, you're not the top market. I think if you look at these instrument sales, the peptide bottom-up proteomics market is I think the dominant driver. So I'd like to be able to say that with like the Human Proteoform project, if I could bring $200, $300 million a year more into the industry and really overcome those challenges, I think in the first phase of the Human Proteoform project, you would get a 10-fold increase in speed. It's the speed and cost. The cost has to drop and the speed has to increase. So please write your senators and tell them to.
I got you with the microphone. Neil, you're fast.
I used to be faster.
Professor, this is Marcia from SciEx.
Hi,
Thank you
Marcia.
so much for this wonderful talk. And the question I have, how do you see the future of all this proteoform studies going into personalized medicine?
I think it's personalized medicine 2.0. Until we get someone's proteoform readout that then you could say, what's the efficacy, what's their disease course, genetics, that genotype to phenotype gap stands. And so that's personalized medicine 1.0. If I have a KRAS spelling error, I want to know about it in my pancreatic cancer. Of course I do. But what beyond that? If you want to know the dosage of a KRAS targeting like a cysteine mutant on the KRAS, if you want to know how much is getting on target, guess the best way, and I think the only way, is to use proteoform analysis to see how much covalent drug has hit your target. That's just one example. But yeah, personalized medicine 2.0, until we domesticate the proteome, it's just going to be elusive. Like, what's really my medical situation? Or could you use serum proteomics at proteoform resolution to correlate or detect disease earlier? It's better information, but that's a giant open question. I would like to say the answer is yes, but we'll just have to see.
Neil, your COVID serum antibodies really got my brain imagining things. And one thing that I imagined was there's so much autoimmune disease in the world these days. And I'm wondering if this isn't a tool to investigate autoimmune disease. One thing that I've been wondering about it for a couple of years is the gut-brain axis, right? So peptides get broken down by your microbiome. And I always wonder, what sample are you going to measure to try and understand the gut-brain axis better? And your example here made me think maybe it's a serum. Maybe we're looking for an antibody in there that is causing an immunocompromised autoimmune type disease, and this would be a way to get to the gut-brain axis mechanisms. Have you given any thought to that?
Yeah, I have. It's why IMPRO has its name. Immunoproteoform landscapes. My daughter had long COVID, as it turns out, and she had a really rough 2 years, and that's why we named the company IMPRO. And she's better now. That was a long road, and it was like a smoldering, tough to diagnose. Sound familiar, right?
Yeah.
Oh boy, it's perhaps one of the more cloudy areas of human biology, and we have just spent a year trying to get cytokine level detection. And as you know, the world's gone crazy with Olink, and we're getting cytokine profiles, but that's protein level, right? We know there's 8 proteoforms of interleukin-8. I remember that because there's 8 proteoforms. And there's cysteine modifying. There's forms of it swimming around. So if you had that knowledge, and we're about a tenfold off in our ability to detect IL-8 in plasma or serum. Maybe if someone's in a cytokine storm, we have enough to detect it. But it's we put a lot of effort into that, and we're still tenfold off. We can see it in PBMCs. If you collect 1 million PBMCs, and we can see some cytokines in there, cytokine proteoforms. So it still needs to get better. I can't say, well, we've got the answer for autoimmunity, but smoldering disease might be able to be detected in other types of proteins in the blood. I'll just leave it at that. And that, it'd be an indirect way to go about it. But I just think the better your information in proteomics, the more you're going to be able to detect.
In this case, you use the immunocapture, and obviously, since it's COVID, you would use something for a spike protein or something like that. But for like the gut-brain axis, how would you choose your target?
I missed his second question. So the pull down, where's the pull down? Yeah, it's a bit later. There. Okay. So the little stars on the bottom of the slide, that's a spike protein, right? There's nothing that says, and maybe we've done this, that you can invert this assay from a standard serology format to putting an antigen, hypothesized antigen, down on the beads and looking for antibodies that bind to it and getting the same single clone resolution data. And I think that may be where you were leading me. So if you put certain bacterial possible epitopes that are really pissing off someone's immune system, and you could do enough of these assays, into the thousands, tens of thousands, to see what really lights up in the serum of a, autoimmune suffering 18-year-old with questionable music choices.
And it seemed to me that it would be possible to multiplex the number of antigens and get a broad array and really go have a high throughput screening there.
Yeah, that's the industrial version, because the spectral space available. Tim, you want to join the board of our company? That would be.
I would love to.
That's an offer. But yeah, I'm showing you. Look at the space of the mass window there. You have, 5, 6 kilodalton up to 150. There's a lot of range there. So you're right.
Tara's got a question. I got her.
Oh, hi, I'm
Hi.
Tara. And,
Good morning.
Good morning.
Karen?
Oh, Tara.
Tara.
Yeah. Sorry, just a really simple question. You mentioned that in older people, the clonality is reduced, and apologies, I don't know the background for that statement. Was that purely from the experiments here to derive that, or is that a common knowledge of that?
We have
within
only
that?
anecdotal. I'd love to
Okay.
show you a careful study design, age-controlled study. It's a secondary index that we have around the patient age in our COVID studies. And but in multiple myeloma or B-cell CLL, it is relatively widely known that as you age, your immune system gets less vital, and one of the ways it does that is fewer clones. Extreme form being one clone that's starting to take over.
Yeah, I guess because in my mind I was thinking if your ability to see clonality is that you have to essentially dilute down your sample, right? You mentioned that in order to even achieve it, you had to reduce it to about 20%, right? To even see those. I was just wondering if you're preferentially looking at high responders, and that if it's just more that you have a series of really low, like your complex of low is just
so
increasing, right? So that your
Fair-
population is more
Fair point.
complex.
Fair point. The degree of clonality, we have to be able to see it. So she's interested in what's below those humps. How many clones are down there, right? And fair point. We probably only have 2 logs of dynamic range in that spectrum, and so maybe there's a lot of low level but mini clones for an older person. But I would still argue the same core message of the talk. That thing correlates to phenotype. Why? Or more tightly than other data types. Why? That's the actual relative abundance of your clones in your blood, swimming around. That's your degree of protection in a vaccine context, or your degree of clonality in fighting antigens. So, but it's true what you say. Maybe there's one clone down there that, if stimulated, there's a 78-year-old with a bacterial infection, and they have plenty of response.
Yeah, I was just thinking, you mentioned the person whose phenotype changed, right? That you had, I think it was, sorry, I don't remember which pathway it was, but that you essentially were expressing one, then it went to not expressing it, and when it comes back for the cancerous pathway, right?
Yeah, MEC1, yeah.
Right.
Yeah, that was with a drug treatment, but yeah,
Right, so it's just, if you had something that, like you said, shot up over a response, it could be that maybe it was hidden before, and now you thought you suppressed it, but it's in that arena, right?
Yeah, you could track someone's, maybe like your question, you could track someone's progression in their immunological status. And I'd love to do that kind of longitudinal study to see if clones come up and down. Maybe what is it, class switching? It's called in immunology. In multiple myeloma, that is one study we're working on. So those are all great questions.
Oh, yeah, it's working. So if there are no more questions, let's give Neil a big round of applause once again.
Thank you.